Hello, welcome back to Godzy. Today we're looking into the sarcomere. The sarcomere is the basic contractile unit of the muscle. In our last video, we discussed about the arrangement or the microscopic structure of the myofibrils, and we talked about the myosin, actin, tropomyosin, and troponin. So today we'll be looking into how these structures, the myofibrils, are arranged in the muscle. All right, so these are the basic contractile unit, and we'll call it the sarcomere. All right, so from the diagram here now we can see the sarcomere. All right, now there are so many structures, there are so many proteins and cytoskeleton proteins, so we'll be taking it gradually so that we understand how the muscle actually functions, how these are arranged in the muscle. So I'll be breaking the sarcomere, the topic sarcomere, into two parts. So in today's video, we're talking about the different zones of the sarcomere, the different cytoskeletal proteins. Then in later video, we'll talk about more of the cytoskeletal proteins and their clinical importance. All right, so now this is a sarcomere. Now the sarcomere is limited by the Z disc. So these structures here are called the Z disc. That's why I had to actually make them in this Z form, so we understand that this is the Z disc. So from a Z disc to a Z disc is known as the sarcomere. So from this structure here, which is the Z disc, down to this Z disc here, is known as the sarcomere. It is actually called Z disc because it is actually kind of a Z shape. So when the muscle contracts this way, when it pulls this way, they distort and form this kind of Z structure. So that's what we call the Z line. It's actually coined from the German word known as Scheibe. Okay, if you want to know, okay, it's come from the German word Scheibe. All right. So the first word Z, we call it Z disc. All right. So this is the Z disc. Okay. Now there are some other structures found here. This rectangular structure is known as the, from our last video, this is the myosin. Okay, so this rectangular structure here is known as the myosin, and these are the heads, the neck, and the tails of the myosin. So this is the myosin. All right, why these blue circular structures here is the actin. So let me quickly label these now as actin, myosin, so that we will know. So this here is actin. All right, so this is myosin. All right, so we understand it very well. All right, so this is actin, this is myosin. All right, so let's look at how they are arranged now in the muscles. Now the myosin is found in an area known as the A band. So they are arranged in a kind of a system known as bands. Okay, so myosin is found in an area known as the A band. All right, so this area that myosin is found is called the A band. So in the A band also, the actins are also found in the A band. So take note of this, okay, because there's so many misconceptions that only myosin is found in the A band. All right, but that's not true, because actin is also found in the A band. So the areas where the myosin is found is known as the A band. So from this area, let me say from here, let's say from here, trace it up here down to this side, up here. Okay, so this is known as the A band. So this is known as the A band. Okay, so we found out that in this A band we can see the actin also in the A band. We can see this actin. Okay, this is actin in the A band. So in the A band we can find the myosin and we can also find the actin. All right, so this is the A band. It is A band because it is called anisotropic. So the A here means what? Anisotropic, because it is the dark portion of the sarcomere. The dark portion of the cell. So if it is viewed under the microscope or under plain polarized light, you find that it is very dark. That's why I shaded it with the red pen. Okay, that's why I shaded it to indicate that the A band is what is the dark portion of the sarcomere. All right, so this is the A band. This is the dark portion of the A band, which includes what? The myosin specifically, and also the actins can also be found in the A band. Now, the area that it is light, okay, the lighter areas of the sarcomere is known as the I band, which means isotropic. I for isotropic. So from this area, okay, down to this area, we will find the actin here is known as the I band. We call here the I band. So from here down to here, okay, we will find the ending part of the actin. So from here down to this part is known as the I band. So here we call here the I band. Okay, I band. I for what? Isotropic. A for anisotropic. So let me indicate that now. Okay, let me indicate that anisotropic. Okay, and the I here means what? Isotropic. All right, so I for isotropic, A for what? Anisotropic. So anisotropic means is the dark portion of the sarcomere, while isotropic means is the lighter portion of the sarcomere. So this is the, from this side, okay, down to here is the I band, while from here down to this side is the A band. Another interesting thing we should also note in the center of the sarcomere is what? You find out that, look at this line here. Look at this line here. So this is known as the M line. The M line. Why is it called the M line? Because it is found in the middle. All right. It is actually coined from the German word known as what? Mittel. Mittel means middle. All right. So this is the M line. So I will call this one the M line. So this is the M line. All right. This is the M line from the German word Mittel. From the German word Mittel. Mittel, which means middle. Which means middle. All right. Now, looking at this M line now, okay, you find out that there are some other brown structures I drew inside this M line. These are the M line proteins. These are the M line proteins. Okay, I'll be coming back to these M line proteins later on. Okay, they are very, very important. Okay, now we've talked about now the I band, the A band, and the M line. Okay, which is the middle. All right. Now, looking closely into this structure, you find that there are some areas that we don't have the actin. Okay, this is the middle part here. There is no actin in this area. So this area is what is lighter than this other areas here. So we say what now? This area from here down to this part, okay, where the actin is not found. Okay, where the actin is not found, we call this area the bare zone or H zone. Okay, bare zone or H zone. So from here, okay, down to here is known as the H zone. H zone. Okay, you can call it the H zone or the bare zone. Okay, or bare zone. You can also call it the bare zone. All right, call it the bare zone or the H zone. This H zone is actually named after the discoverer Helsing. Okay, that's why it's known as the H zone Helsing. Okay, it's also coined from the German words also. All right, so I don't know why they're always coining now most of the names of these structures from their German words. All right, I think the German word means something hell. Okay, you can check it out yourself. All right, but it's actually named after the discoverer Helsing. So the name the H zone. All right, so this zone that doesn't do not have the actin filament is known as the H zone, and in the H zone they have also the M line. All right, now let's talk more about the Z line, the Z disc. Okay, so this is a Z disc, and as I said earlier, it is coined from the German one Zwischenscheibe. Okay, Zwischenscheibe. And looking at this Z disc here, we find out that there are some proteins there's a protein that anchors each Z line together. Okay, these Z line are held together by a protein. Okay, I didn't indicate this protein in this structure. I only indicated it in the ending part of these structures. Okay, this protein is known as desmin. Okay, this protein here is known as desmin. So I will call this one desmin. This is desmin. All right, so this is desmin. All right, we'll talk about more about these proteins later on. Okay, so here we have the I band, the A band, the H zone or base zone. We also have the what? The M line. And remember, the I means what? Isotropic. A is for anisotropic. So let's watch closely now. If you look at the myosin, looking closely at the myosin, we find out that what there is a cytoskeletal protein that is running through this myosin. Okay, it's running through it and attaching what to the M line. Okay, and it's coming off here. So this protein here is known as titin. Okay, it's known as what? Titin. All right, it's known as titin. All right, titin. All right. So the function of this titin, okay, is to anchor the myosin to the Z disc. So the titin is an elastic cytoskeletal protein. So that's why I drew it in what? In this spring-like shape, because it is elastic. Okay, when the muscle contracts, it actually helps the muscle to what? Retain its or come back to what? Its normal shape. Okay, so it is elastic in nature, right? So it anchors the myosin to the Z disc, and also what? Anchors it what? To the M line proteins. Okay, to the M line proteins. Now it has two terminals. It has the carboxy terminal and also the amino terminal. The terminal that runs in or through the myosin is known as the carboxy terminal. So this is the carboxy terminal of the titin. Why here is the amino terminal. So let me indicate that now. So here we have the titin. Here is the amino. So here we have the amino terminal. Amino terminal. All right. So why the one that runs through it is known as what? The carboxy terminal. The carboxy terminal. The carboxy terminal. So all right, let's move now into the M-line proteins. Now let's talk about the M-line proteins. I say what now? That the titin has a carboxy terminal and an amino terminal. The carboxy terminal is the one that runs through the myosin and attaches what to the M-line protein. So it anchors the myosin to the M-line proteins and also anchors the myosin what to the Z disc. All right. Now looking into what the M-line. Okay, we have the M-line proteins, and these M-line proteins what include obscurin, myomycin, M protein, C proteins, and creatine kinase. These are the M-line proteins. So the M-line proteins, what will I name them now? Okay, M-line proteins. So these circular structures I drew in this brown color are known as the M-line proteins. So these M-line proteins include, all right, I'm just going to name them here now. They include myomycin. Let me use the blue pen, the black pen, or maybe red. Okay, so that we can differentiate it very well. So we have what? The myomycin. Okay, we also have the obscurin. We have the C proteins. We have the M proteins. We also have the C proteins, M protein, obscurin, myomycin, and we have the creatine kinase. Creatine kinase. So these are the M-line proteins. These are the M-line proteins. Now let's talk about the importance of these M-line proteins. Myomycin helps to stabilize these this myosin. Okay, it helps to stabilize this myosin to keep them in their spatial relation. Okay, it helps to what? keep the code. Remember, I said that what the titin attaches what to the M-line protein. So it attaches what to this M-line protein, specifically to the myomycin, okay, that it helps what to stabilize their relationship, okay, so that what it helps to keep their spatial relation to each other. All right, so that is the function of myomycin. So obscurin also helps maintain what the structural integrity of these whole myofibrillar system, okay, that's the function of obscurin. All right, so C proteins and M proteins are very important in this whole myofibrillar system. Creatine kinase is important in, I think that should be under the metabolism of muscles. All right, so creatine kinase, all right, is not a structural protein, just like unlike myomycin. Creatine kinase is more like an enzyme. All right, what creatine kinase does is that it takes a phosphate group from creatine phosphate and it phosphorylates ADP into what ATP. That's the function of creatine kinase. All right, so this ATP it's used by the muscle for contraction. All right, so this is a way that the muscle actually develops its own ATP for muscle contraction. All right, so that's the function of what this creatine kinase. So these are the M-line proteins. Okay, let's go over it again. M-line proteins include myomycin, obscurin, C proteins, M proteins, and creatine kinase. So these are the M-line proteins. All right, so what really happens there is titin what the carboxyl terminal of titin runs through the myosin and attaches what to the M-line proteins. All right, and the amino terminal attaches what the myosin what to the Z disc. All right, so this is the M-line proteins. All right, let's move to the actin. So looking at the actin here, we find out that what there's a straight structure, okay, or protein running through this actin. All right, remember in our last video we talked about the structure of the actin. All right, so I didn't bother putting all those other structures of discussing. So if you've not watched or seen the video on the structure of actin and myosin, please, I'll leave the link the link to the video in the description down below. Okay, so make sure you watch the video. All right, so let's now go into the actin. Now, looking at this structure here, there's a red indicated it as red, a protein running through the actin. This protein is known as nebulin. It's known as what? Nebulin. All right, so this protein here is called what? Nebulin. All right, so nebulin is known as the molecular ruler of the actin. All right, it is inelastic, unlike the titin that is what? Elastic. Nebulin is inelastic, so that's why it is called the molecular ruler of the actin. So it actually set the shape or the length of this actin, so it helps the actin keep its length. All right, so not to extend more than its length, so that's why it is called the molecular ruler. All right, so that's the function of this protein known as what? Nebulin. All right, so now looking at this nebulin, the nebulin also what attaches what this actin what to the Z disc. All right, it attaches what this actin to the Z disc. All right, remember this one here, this green colored protein here is known as the Godzy. All right, this is known as the Godzy. So Godzy is a protein that actually covers up the positive end of the actin. So this is the positive end of the actin, while here is the negative end. All right, so this is the positive end of the actin, while here is negative end. All right, so this is the positive end, negative end, positive end, and negative end. So let me just indicate it now: positive end and the negative end. So at the positive end of this actin, a protein known as what? Godzy covers what? The positive end of this actin. Why? All right, I will tell you later on. Looking at the negative end, another protein what? Known as tropomodulin covers the negative end. So this protein here is known as what? Tropomodulin. So this protein here, this black protein here, is known as what? Tropomodulin. Modulin. All right. what tropomodeling does is what? Is to prevent the what? The additional attachment of what? G-actin. That's the function of tropomodeling. All right. Remember, in our last video, I said what? The G-actin, which is the globular actin, is always polymerized into F-actin. Now, if tropomodeling do not what? Covers what? The negative end of the actin, more what? G-actin will be what? Will be attached to these what? Actin. And remember, we have to keep what? The length of the actin what? The same way. All right, the same way. That's the function of the nebulin, so that what? We know the length of the actin, because if the length of the actin increases, remember, contraction will be very, very weak also, because now, if assuming we increase the length now of these actin, so when the module contracts and pulls this Z disc and this actin towards the midline, all right, push it towards the midline, find out that what the length of contraction will not be equal as the force of contraction will not be equal as when this actin is shorter. All right, so that's the function of tropomodeling. All right, now tropomodeling covers the negative end to prevent what? An additional attachment of what? G-actin. So that's the function of tropomodeling. Why the Godzy covers the positive end and also attaches with the edge of nebulin, attaches it what? To the Z disc. All right, so this is the actin, and these are the proteins that function what? In the actin. Remember, oh, I forgot to say something about the Z disc. All right, now looking at the Z disc, I've mentioned a protein that holds the Z disc together, known as desmin. All right, now looking at this Z disc, there are these important structures I drew here. All right, looking at them here, these important structures I drew here, these are known as what? Alpha actinin. Please don't confuse alpha actinin and actin. They are different what things entirely. All right, so this structure here is known as what? Alpha actinin. Alpha actinin. What alpha actinin does is that it connects what? The actin to what? The Z disc, just from its name. Alpha actinin connects what? The actin. So this whole system, both the nebulin, the Godzy, all right, and everything, it connects them to the Z disc. So the proteins of the Z disc, remember, the protein of the Z disc are what? Alpha actinin and Godzy. So these what? Are proteins of what? Of the Z disc. Okay, Z disc protein. Z disc protein. So the Godzy and the alpha actinin are what? The Z disc protein. All right, alpha actinin connects the actin what? To the Z disc, while the Godzy covers the positive end of the actin and also assists what? In the attachment what? To the Z disc. All right, so we've covered most of the proteins here. All right, we've talked about the desmin, all right, that helps what? Anchors Z disc to each other. All right, we talked about the amino and the carboxy terminal of the titin and what the function of the titin. It connects what? The myosin what? To the Z disc. We talked about the actin, the tropomodeling, and the Godzy. All right, we talked about the alpha actinin that connects what? The actin to the Z disc, and the nebulin, the molecular ruler that keeps the length of the actinin. So we also discussed here the M line, all right, and we talked about the proteins of the M line. The proteins of the M line include the myosin, obscurin, C proteins, M proteins, and creatine kinase. So I've talked about the functions. Myomycin, I said the function of myomycin is what? Is to what? Connect the carboxy terminal of this titin also, and also keeps the spatial relation of these myosin molecules. All right, I said obscurin helps to keep what? The structural integrity of the myofibrillar system. All right, and I said that creatine kinase helps what? Phosphorylate ADP into ADP by taking off phosphate from creatine phosphate. All right, I said desmin here helps what? Connect Z discs to one another. All right, so this is the sarcomere. All right, so in later videos we'll be talking about other structures you can see here. All right, these structures, other structures you can see here, we'll be discussing them in later videos. So if you really enjoyed this video, please don't forget to subscribe to this channel and also leave a comment in the comment section below, and don't forget to share this video with your friends.